Om Amriteshware Namaha. Good afternoon to all gathered here. I am Dr. Aishwarya Sudan.
And I'm Dr. Lakshmi J. We are junior residents in Department of Pharmacology, Amrita Institute of Medical Sciences, Kochi.
I would like to welcome all of you on the Zoom platform to engage in this dialogue on animal experimentation and its relevance today. Humans need to be humane for the sake of humanity. Today we are commemorating the World Health Day, the theme of which is Our Planet, Our Health. This webinar juxtaposes the minimalistic use of animals in accordance with this theme of the WHO World Health Day program.
Let us invoke the blessings of the most benevolent Amma, whose grace pervades our institution.
With our appeal for holy blessings, I would now like to request Dr. Princey Louis Palati, the head of the Department of Pharmacology, Amrita Institute of Medical Sciences, to welcome the gathering. Over to you, ma'am.
My humble pranams at the lotus feet of Amma. Experimental drugs, 9 out of 10 fail in clinical trials. We have very many laboratory and animal experiments that lead to potential molecules that is there. But we always feel that with the rise and advancement of technology, why should we otherwise go into animal studies still? Can't technology like 3D printing, also 3D models, we have also biochips, we have stem cells, can't they all release and refuse to use animal studies in this day and age? This is a very big question. And as a pharmacologist, as a new crop of residents come from the institute, we have to deliberate on the relevance of animal experimentation today. How to do it is a different thing, but what is the state of animal experimentation today? So, friends, it is said if I were to talk positively for animal experiments, I would be having a whole animal rights activists hurting me and my family for my positive aspects in this. I just would like to state a quote from some place that they have said, animal research, stopping animal research, is that the killing field, or if we stop animal research, are we stopping the real killers? So this is a point that we have to think on. So animal experimentation should be, you know the formula, the 3 R's and the 4 R's and the S and the ABCDF, but I should like to tell you that animal studies are here to stay, but we can reduce it to the maximum. We have to use it responsibly. Just like how you use social media, it's a good thing and a bad, internet, darknet, everything can be used responsibly for the betterment. When COVID came, what happened? We did not have a drug. That was the main cause for immediate anxiety. But then we did, and now we are without mask, we are getting out of the COVID. It will be there, ever present in a small area, but that is how it is. We conform, and for that we need to have drugs. And as a pharmacologist into research, there are many of us who have to know how to do animal experimentation as and when the need would arise. And that is the reason, friends, we are here to deliberate on this topic on animal experimentation, its relevance today, as our very important and distinguished speakers are here. So, with much ado, I would like to welcome our director in absentia, our principal, Dr. Colonel Vishal Marwaha, for having accepted and allowed for such positive ventures always in Amrita Institute of Medical Sciences. And we have with us a very distinguished panel of speakers, and that exactly goes to show the minute the brochure came out, we had 341 registrations within 24 hours, and right now we have about 695 registrations, so to speak. So you know the topic is relevant, and we have distinguished panel. Like I would like to welcome Dr. Vikash Medhi from PGI Chandigarh, Dr. Ajay Prakash, and Dr. R.K. Dixit. He's in absentia, he's out, but he will be listening on to us. And being the true professional that he is, he has sent a video because he can't join online. He's in a flight for emergency commitments that he has to go to. And I would also like to invite a young entrepreneur, a medico, Dr. Manasa, too. So this is a small yet a very condensed capsule-sized deliberation on animal experimentation from which you shall benefit a lot. And I also welcome my department, my dear students of Amrita Institute and others, and our Dr. Pg Nair, who is the epitome of pharmacology in Amrita right from its inception. And we have our beautiful MCs, Dr. Lakshmi and Dr. Aishwarya, and my team with Dr. Sanita and Dr. Danya, and our Mamta, Vandita, Rosmi, and Dr. Dipti, all together within this. So welcome all. You are in for an exclusive deliberation on animal experimentation.
Thank you, ma'am, for your wonderful take. Animal studies are really here to stay. Like social media, it is truly up to us to use it for good or for bad.
I request our beloved principal and the dignitaries to symbolically inaugurate this function by lighting the virtual lamp. As the light of a lamp dispels darkness, so the light of education eliminates the darkness of ignorance.
I am extremely honored to introduce our chief guest for the occasion, Dr. Colonel Vishal Marwa. Sir serves as the current principal of Amrita Institute of Medical Sciences and is professor in the Department of Rheumatology, Amrita School of Medicine. Sir has completed his training in rheumatology from All India Institute of Medical Sciences, New Delhi, and Nottingham University, United Kingdom, followed by an international scholarship from Japan University in 2006. On a personal note, I would like to add our principal is approachable and amicable, especially to students and peers alike. Over to you, sir.
Dr. Pg Nair ma'am, Dr. Princey Lewis ma'am, Dr. Sanita, all the faculty members, guests, and students attending this function today. Use of animals in education and research purposes, very debated. Killing of animals at school and UG levels, they say, may not be ethically justified when computer software for the same are available. But there are situations where one has to do animal experimentation. I think this will be basically there'll be a lot of deliberation on this subject, and I must admit that we have stalwarts in the fields: Dr. Vikash Medhi, Dr. Rakesh Dixit, Dr. Ajay Prakash, Dr. Manasi. All of them will be speaking, and I'm sure that will improve the knowledge quotient of the subject. So I think in such a scenario where there'll be a lot of deliberations, I think it's better to speak less. So I'll close for now and wishing all the best, and I'm sure that all the the speakers and the attendees will benefit a lot with the deliberations. Thank you very much.
We shall now enter the scientific session by welcoming our first speaker for the day, Dr. Vikash Medhi, who will be speaking on animal experimentation and its relevance today. Sir needs no introduction, but I am privileged to declare his achievements. Sir is serving as Professor and Additional Superintendent at Department of Pharmacology, PG Chandigarh. He is also the coordinator of Pharmacovigilance Center and Regional Resource Center for the Northern Region for training and technical support. Sir is the recipient of many prestigious awards, such as the Dr. D.N. Prasad Memorial Award in 2009 from ICMR, New Delhi. Over to you, sir.
So thank you very much. I think actually it was difficult for me to, get the internet. Anyway, so what today, since today is a World Health Day, and as ma'am has already elaborated what is the importance of animal experimentation, also Dr. Marwa, what he said is with his experience from undergraduate, while he was doing during that time number of experiments, and today's if you look at the curriculum, what are the changes is there. So let's take a balance. I'm going to speak about the importance of animal experiment and what are the alternative and how we should go in future. That is important. Now if you look at this, that all these advances, whatever you are getting it today, it is based on the experimental studies, and that what it has been translated in terms of the drug development or in terms of a new treatment modalities also. It was actually initial data has been generated. It is from the animal experimentation, and that is why we say that we are this important. I'll just. So what if we're referring to some of these, very renowned person who are actively involved. So all you get it, my milestone, whatever you are getting, it is because of the animal experimentation. But if you look at specifically also that what we developed so far in number of decades, if you look at life-saving surgical procedure, if you look at laparoscopy, we learn it from people used to go to Germany and other places. Our surgeon is initially with the pigs. If you look at the cancer therapy, any of the transplantation we talked about, recent experience with the vaccine, what we develop, normally it takes 10 years, 12 years. Still we're looking for a vaccine for HIV, it is almost 40 years. But we could develop the vaccine based on the animal experimentation within 8 months. So there are several examples for safer consumer product or the treatment what we develop for any of the medical disorders and disease. But coming here is, some of this issue raising by different stakeholders that should we go for a conventional animal experimentation in teaching or during the research also? Can we have an alternative? So if you look at this paper, which is published in Nature, that India is trying to look for an alternative to animal in biomedical research, not only drug development, in diagnostic and other areas also. So if you look at that, there are some few has been put looking at computer simulation, what we do normally in silico technique, or you can extrapolate this information into the technique. And if you look at that a couple of years back, what we are trying to develop is that normally when you do a regulatory toxicity study, it is going to take 5 years time. So people are trying to go for a micro-dosing studies so that you can give a minor dose and see that what is a human behavior. Of course, when you look for an alternative, that need to be validated. And that is why in a drug discovery process, if you wanted to override that, you need not have to use the animal experimentation and other method. So definitely it need to be validated. And then only, of course, with the regulation over a period of time, we are judiciously use the animal in animal experimentation. So these are the view from the, it was published in Nature, and some of the scientists they expressed their view on both sides, what will be the alternative or if there is an alternative, can we accept, or is there any regulatory acceptances there? But otherwise, if you look at, number of example that in terms of, developing vaccine for leprosy, or number of studies like we refer it to AIDS. So all the smaller animal and higher animals we have been using, and we are getting benefit from, from the data. So here one point I like to highlight: if you happen to go to any developed countries, they are, doing all the experiment. If you look at today's, if you ask a question, how many, in an academic or in the industry, we have a facilities for monkey, you can just count 1, 2, or 3. How many facilities we have a Beagle drug, you can count it, because fortunately, we are the Oecd approved around 50 labs. But over a period of time, like last year, one of the lab has started a mini pig where it was not there in India. And since we do not have a monkey, some of the institute like ICMR Institute, they use a marmoset. So I think the facilities should be available, and we need to make it, because if we do not have it, then definitely it will interfere with the drug development process. And that is what we are looking at. We are very strong at formulation, but when you look for how many institutes have a new chemical entity, have been done in a new drug, and that we have count 1, 2, 3, 4 like this. So that is why the question it is asked, that when you do animal experimentation, that is it you are unnecessary doing, or you have a justification. So we have a justification that unless you generate the data, if you take an example of thalidomide, it was done in mice and rat, but it was done also in mammals, and they hide the result. So that is what we get to learn, that there is a species variation. Now we understand the individual variation. So that's how we learn from this. So the question here, if you ask that why all this animal experimentation, if you take an example of smaller animal and higher animal, it has been done. So we have number of justification, because, if you look at the similarity, or if you look at it, that you can extrapolate maybe 30 to 40%, but at least you have a, you can address, unless you do a live or in vivo, you cannot address biological possibility. So these are some of these, point need to be looked at, that how it will mimic in a human condition, how much it is mimic, can you identify biological possibilities? That is why. So normally when you do experiment that in academic or industry, you totally, if you see that more than 70% it is we use mice, different type of mice, different type of rats. Of course, we have rabbit, guinea pig, and other animal also, depending on the research. But availability. So for PG student, if you do not use the animal, I have seen in number of examination, if you ask, they did not see the mice, they do not have a facility, and it becomes difficult. So over a period of time, if we see that we will not have any skilled manpower for any experimental research. So many of the people like PG students, they might think that they want to become a clinical pharmacologist, but how do you become a clinical pharmacologist if you do not know how to establish the regulatory, data, which will be using for an IND application for making a phase 1 protocol, and how do you mimic, like suppose if you do go for a kinetic also, how you're going to mimic in a human condition and to write a protocol for phase 1. So that is why that animal experimentation is required. If you look at the conventional one, that unless you have an alternative which is validated, which is acceptable by all the regulatory authority, like if you look at ICH region regulatory authority or Indian regulatory authority, before you initiate a human clinical testing. So now, what is our infrastructure? We have 605 medical colleges, we have 22 AIMS, we have bigger institute. If you look at ICMR Institute, we have 34 institutes there. And one more important point I'd like to highlight, it was about to be inaugurated on 2nd April, and it was an institute for catering all the need for animal experimentation, and it was built by government in Hyderabad, one of the latest ICMR institute. So any of the species is not available, like we get a cell culture from Pune. So all the scientists, so this is one thing has been done for the last 5 years. So in order to cater to, in order to, create an infrastructure for all these academic institute or industry. So this has been done for a scientific organization. So if you look at the infrastructure, I talked about medical college, major institute like Amrita, or if you take about example of 38 CSR lab, so including the CTRI. So these are the infrastructure we have. But taking, utmost care, if you say that you do not have a facilities, because if you look at the CPCSA guideline, you cannot maintain the standard. And that is why, though people say animal experiment is not allowed, but the question here is that institute cannot maintain with the CPCA guidelines. So that is why. So we need to be trained the people. Of course, we'll talk about ethics in the second lecture. So that is why. So otherwise, if you look at all together, it is very important to know the initial finding, so that you get an indication that, you talk about that you wanted to use a drug for 10 days, so you have a acute toxicity data, and how you're going to respond if you say that, you wanted to use the drug for 10% of the lifespan, that we have repeated those toxicity study, whether drug causes mutagenicity, carcinogenicity, you have a long-term toxicity study. So that is why you need to generate the data. Unless you generate, you cannot justify for going for a clinical study. Like, for example, if you happen to go for a clinical study, phase 1 study, and you have a healthy volunteers, male and female. So if you do not do a reproductive toxicity study, how do you justify? How do you justify for using this? So that is why you need to know that all the information for this information should be required. So we need to have an alternative one. At the same time, what I'm trying to tell is that if you can justify, you can develop a method which can be used for alternative, like we use for some of the cosmetic one. So that is good. Like one thing should be also remember, whatever we do the research in animal, it is also, we can see that how many veterinary product is also developed. And we use for animal welfare also. All these product, all these drug we use for. So that point is also, so mutually get benefit for human or also for animal also. So we have number of benefit if you look at that drug we develop, we use for veterinary product also. So that is why if you look at that animal experiment is unavoidable. You cannot replace the whole totality, but what I'm trying to tell is that if you can develop an alternative method which is acceptable, then you can say. So you can take an example of thalidomide. We could have avoided 10,000 baby born with a phacomalia. But only thing is that time regulatory was not stringent, like today's era. So take an example during COVID, what we learned. Please refer to one of the article also we published in Virology Sinica, which is published from Wuhan laboratory. What is the ideal model for, COVID-19, like this kind of viral infection? So if you look at that, in developing vaccine also, they have to go to the forest area to look for a rhesus monkey. Okay, so you can say that there are some models which is ideal for a particular disease. We have number of examples. Many times we do try to develop a disease, but animal is resistant, and that is why this information or data is very, very important. So you can learn a lesson from during the COVID, how different models were used to develop the vaccine. So if you look at some of the survey which is done in US, if you look at 95% of the animal, some of this animal, it is not included. But if you look at some of the survey, like 89% scientists survey, the peer review research, the animal testing research, or if you look at higher animal, that how it has been used 99% for, which is almost similar to human, 99%. And even if you look at the mice, 98% genetic similarity is there. So all the developed countries like US or other European nations, they extensively use. So here there is a number of examples I have quoted, so you should know it. So one thing is that if you take an example that we do not use a frog, if we're looking at teaching earlier, we wanted to look at that heart-lung preparation or circulation, or we wanted to use the panel of drug. But nowadays we do not use because we thought that it is affect the environmental. But otherwise, if you look at the data, how many millions of rupees we earn by exporting countries like China and other the frog. So that is also you need to know that if we're looking at that you are not going to use frog, I'm not justifying, but we are also exporting for making a money. So there are a couple of recommendations if you look at that regulatory authority or a global guideline or Indian guideline. So ultimately we wanted to have a refinement on the experiment And please go to the ARRIVE guideline also, CPCA guideline also. We should understand that what is a guideline means. It is attached to Animal Cruelty Act, so it is almost become a mandatory. So that is why we need to have one more positive point is we have 50 lab in India, which is Oecd. I'll talk about detail. And that is why all that when you plan for any studies, how do you design, analysis, statistics, and in the form of publication, it is important. So question need to ask that, do we really have a right to do experiments? That is what. So one justification I said that we do not have a full-time replacement, and also it give a mutual benefit. So with this guideline and regulation, we know that we have CPCA guideline, ARAB guideline, then Iaec, so many guidelines are there. So this is to streamline or need to justify so that there is no duplication of the work. If somebody had already done it, why you are going to do it again? So that is why adequate data should be there, which can be, with this data, you can take a decision for next step two in a drug development or for a diagnostic also. So these are the things. So of course, Dr. Ajay will talk about detail of all principle. We need to know that when we do experiment about 3 hours, but some of the hour we also take it at another hour, 5 hours. We should also take it a responsibility. So we put it in a books for more popularity of, So when you talk about alternative, like you say, can you replace the in vivo situation? Like if you say, we can do in living situation, like, cell culture, or if you have an isolated tissue, or you have a human base. Suppose you wanted to say that you wanted to see that effect of the drug on any vessels. So if you can take human vessels, you can do it. That is much better, or brain tissue. So from the surgical excision, we can do it. So, but if you do not have, at least you can generate the data in vivo situation. We can also do a non-living situation, like we have a computer simulation, but the question still remains that the biological possibility. You can do a lot of imaging technique or DMA recommended mathematical model. We have several methods by non-living technique is there, but only question is, may not be possible to extrapolate 100%. And even if you can justify, even if you do in vivo study, maybe 30-40% it is simulating, or you can say that maybe if it is safe, you can go for the next phase. So we have many procedure, like we say, if you look at the regulation, if you go for long-term toxicity study, maybe 2.5 years, but if you go beyond that, definitely you have to justify. So all you talk about, look at that, our guideline, period of restraint, training. If you happen to go to any university, you wanted to do animal experiment, at this stage also, if I go, I have to undergo a orientation program. Though I might say that I've been handling animal for last 20 years, but this is what the SOP is required. So that is what it is doing. So we should tell the PG student that, how the experiment should be conducted, all the regulation, and then only you can have a skilled manpower for the future. So that is why even if you do not have a higher animal, at least you can have a smaller animal, differentiation, justification, why you are using this animal for what disease, what is the output, what is the ideal animal, everything should be talked about. So normally when we go for the examination, when we ask that, can you tell me the name of the mice? They might say, albino mice. If you look for it, can you tell me the strain or different name? What kind of experiment you are going to do, a Balsi mice or C57 mice? They'll not be able to tell you. This is because that we have a limitation in a curriculum or non-availability of many of the peripheral medical colleges. I think that need to be overcome, that they should be trained. Maybe one more important point I can refer that one of the meeting I had attended, aspect meeting, where there is a big publication in a peer-reviewed journal, whatever funding has been given, this data cannot be reproducible. So one more issue that when you plan for any study, you need to look for that, is the design is adequate? And that's why you need to train. And in this meeting, NIH actually said that we'll give them money for training the people, how the design should be planned, how the study should be conducted, how you're going to randomize. If you do not have a software, how do you do it? So these are the points need to be highlighted for a PG student. So if you look at some of the key coordination or key point for a futuristic approach, that we know that there should be restriction of animal experiment. But if you do not make it available, how you can develop a skill manpower for future. Suppose today I'm working for 20 years, my PGs are not seen, then we have to depend on outside. That is what is happening actually. If you go for some of the point that why there is a pressurized, we are following the guideline. We should also develop expertise for alternative like in silico or other technique. Definitely it is acceptable. So you have number of parameters you can evaluate, then it verify by in vivo technique. So altogether, if you look at the final point, using animal in research, it's a privilege. And this privilege with the regulation, with the self-responsibility should be there, that how they should be used. At least a human, when you handle the animal, human, how do you handle in a clinical trial should be there in animal experiment also. So otherwise, you can see technology. We want a new technology where it can predict. Like we have a in silico method. We have different softwares are there where you can say we have a high put through. So this only need to know that when you go for a drug development, suppose you do several tests or parameters in terms of acute toxicity, repeated dose, or long term or safety pharmacology. Definitely, we have to. We cannot, supplement with in silico technique. That has to be done in the animal experiment also. So if you look at the number of experiment, I have already shown the slide. Mice is the number one, then the rat, then all the higher animal. But if you look at number of study in the field of pharmacology also, majority is for drug development. If you look at Indian infrastructure also, Oecd lab, we have 50 lab. They have been doing a toxicity study for all parameter, acute, repeated dose, or long-term toxicity study, or safety pharmacology study. But otherwise, conventional proof of concept studies, bioequivalence, okay, all these studies you can use the animal, and that is what needed is done. So look at some of the scenario, how the scenario is changed. If you look at 1960, 70, you used to work for prostaglandin. Later on, we work for endothelin. Now, if you look at last 10, 12 years, people are looking for a nanotechnology or nano product or the stem cell. And that's how we could see that, 1980s, what are the, different aspect we do, 1990, but still we are looking for an alternative to animal experimentation. That is what, maybe futuristic approach, but what I say again and again, it needs to be accepted by regulatory authority. Now, as you can see that during COVID, what we learned? We learned many things. So during COVID, if you see that how different experimentation has been done, like many of this industry are affected, but many are proactively it was done during the COVID. So you can see that different countries, like in Germany, how during the lockdown, different experiment has been done during this time in different animal, smaller animal, higher animal. So this is a proof, is a kind of that all the developed countries, they are actively participating in the drug development or in the academic setup, also training the people. So that is what we need to learn, that if we do not do it here, so tomorrow we'll end up with the people, we do not have any expertise. Anything you wanted to do, we need to, we do have to take a comment from these developed countries, some of the experts, because we will not have anybody to lab. Because if you do not do it today, tomorrow we'll not have any next generation. So that is what we need to do. So any of this approval of investigational new drug or any of this new drug, what you wanted to evaluate as toxic, how much it is, what dose it is, safer to use in a human condition, how you're going for a screening. So these are the point, and how you identify some of the risks, that only you give when you do the study proof of concept on all the regulatory studies in the smaller and higher animal with the different species. So we need to tell that student how you identify that you need to train for the in silico, or as new branches coming, if you look at that, lot of gray area of medical devices. So that also you need, and even in nutrition or any of the nutritional product, we also do a regulatory toxicity study, and including cosmetic. Of course, in cosmetic, we have some replacement with animal studies, but that also need to be looked for in near future. So I think you have a long way to go, but moral of the story is that whatever you do, your data is accepted if you say you do under OECD principle, Organization of Economic Cooperation and Development. As I said that India, we are the vice chair, and we have more than 50 labs, but all the academic institute, even if you are not GLP, you try to make it a mimic to the GLP, you follow the principle of GLP, and that's what we need to do in future. So when we talk about the regulation, that you talk about NMC, the new name for MCI, that they regulate, and how you're going to train the undergraduate student. Of course, now it is, whole thing has been changed, but still we need to know that even that, pharmacist institute or different university, so we have the regulation from different regulatory authority. So those who like to read, they can read that some of the guideline like CPCSA or ARRIVE guideline, please go through it. So you can be more familiar with how you should do with the regulation, that it is not going, nobody is stopping that animal experiment. Only thing is that how you are going to do it, how you are going to keep the standard, that is important. So with this, I just summarize that my point is, I wanted to make a balance. People who are for animal experiment, people who are against the animal experiment, we need to make a balance. Why the balance is there? Because unless we develop a validated or global acceptability method for any of the development or any of the identify risks and toxicity, so it cannot be implemented. So whatever conventional way that we have a regulation, we must generate so that we can, we have a robust data, we can say that we can go to the human condition or clinical trial based on the animal experimentation. Of course, earlier when you look at that, now only PKPD data in animal experiment we can develop, and it is going to help in a phase 1 study and phase 2 study. So these are techniques should be applied. And if you have alternative one, you can do a battery of test also. That is also beneficial. Thank you very much. Thank you, everyone. So if you have any question, I'll be happy to answer.
So there's a question. So can we go postgraduate work? Praveen is asking. So when Mr. Praveen is asking, sir, can we use this for postgraduate teaching?
Yeah, actually, there is a regulation that when you plan for any of the experiment for PG, because it is in a curriculum of MD Pharmacology, so or MSc Pharmacology, what you can do is you can approach the Institute Animal Ethics Committee, number of experiment you are going to do, and you get an approval, and then you can start. That is there. So you have to get an approval from Institute Animal Ethics Committee. So the provision is that any hazard chemical you use, initially IBC clearance, or second is Institute Animal Ethics Committee. For higher animal, you have to get it to CPCA headquarters. But any of this training program, you get an approval from Institute Animal Ethics Committee. That is there.
So there's one more question. Is it mandatory during NMC inspection during PG Pharmacology Inspection?
So it is in a syllabus. So if you do not have, you can have a simulation, because many of the institute, they do not have a in vivo situation. So there also it has been accepted, but those who are not able to maintain that, maintenance of animal as per CPCSA guideline. So this is accepted, but it's better to have it. Otherwise, you will not have any expertise, because if you do not teach, do not train, how they are going to get the training.
Is there any further queries? So there's one more query. Could you please tell something about animal simulator softwares?
So these are used. One is Elsevier, where basically people see that we train the people that if we use heart-lung preparation, for example, so you have a panel of drug. We have adrenaline, noradrenaline, or inhibitory drug. So that's how you look at that autonomic nervous system. Second thing is for PG student, we have a preparation is carotid. How do you record the blood pressure? So we had earlier the dog BP, then cat BP. So this actually shows that when you isolate the carotid, one you calculate. So there you make a simulation that if you give a normal saline, what is the volume effect? Okay, then when you cannulate, how it is going to increase the blood pressure. So if you block both the carotid artery, if you lift the carotid artery, how it is going to increase the blood pressure. Then you give adrenaline, noradrenaline, like panel of drug. So this is to basically to make you understand that how the different drug affect the blood pressure regulation. So that's how this simulation technique has been used for any of this experiment.
Sir, there's one more query. So could you please repeat what are the 4 species recommended by WHO for COVID-19 experimentation, and why specifically those 4 species, sir?
See, one thing is that in anything you wanted to go for a immunologically mediated action, because in COVID you need a immunomodulator. Okay, so that is like, for example, if you do not have anything, then you go for a bulb C mice. If you have a availability of albino mice, Swiss albino mice, you're not going to do it in Swiss albino mice. You're going to do it in bulb C mice because that is more relevant. So if you have a hamster, then I'll prefer to use a hamster. So like that it is there. So I request you to read, or otherwise I'll send a paper where we publish that what is the ideal model for a COVID. We publish in Virology Sinica. So we'll put it in a mail so you can go through it.
So there is one more question. How will we receive approval from IAEC for PG course animal experimentation, as all those experiments are repeat use?
No, this is a provision that we practice in our institute, that like 1 year, how many experiment you do, like PDZ induced, MES, like that, or Bioassays. Okay, so there you take a minimum one, because one animal is sufficient to use for, you go for intestine or any of the stomach. So there you put it, these are the experiment you are going to do it, and you apply to with the form B to Institute Animal Ethics Committee. So they'll give the approval. Once you get an approval, so every year you have to put also annual report. This provision is there. You can do it.
Sir, the next query is, can you please explain what are the ARRIVE guidelines?
See, like in India, we have many guidelines. One is, if you say, we have an INSA guideline, Indian National Science Academy for Animal Experiment. Okay, we have a WHO guideline. We have a guideline from ICMR also, if you look at the earlier one. But we only follow the guideline, which is mandatory, is CPC-SA guideline. So this CPC-SA guideline is mandatory because it is attached to Animal Cruelty Act. So anybody do a regulatory toxicity study or any study, proof of concept study, we have to say in a protocol that you are going to follow the CPC-SA guideline. Similarly, global term that, we have a ARRIVE guideline. So you talk about that any particular group, how many animals you are going to use, what should be the condition, how you are going to maintain. So these are the global guideline, ARRIVE guideline. Or any experiment, what you say that when you are going to do it, I also request that you are going to do it under OECD principle. That is Organization of Economic Cooperation and Development under GLP, Good Laboratory Practice Guideline. So ARRIVE guideline is a global guideline, you can follow it. So anything like, suppose you do clinical study, you say I follow GCP. At the same time, I follow ICH GCP also. Because if you say that I wanted to submit this data to any countries like US, Europe, or other, so then you have to say that I have done the study under the global guideline, I follow it, or under OECD principle. So then you can submit the data to any of these, regulatory authority. And that is why just read that ARRIVE guideline along with CPC, you can have a comparative also assessment that you can do. And these guidelines are regularly updated. That also you should. Yeah.
So there is one more query. Where India stands in animal research?
See, I already told you that India has a one good point is we are the vice chair for the OECD. We have 50 plus lab. When you compare to China, that we have an advantage. But otherwise, if you see that most of the academic institute, medical college, we are not maintaining the animal research. So that is also alarming. Over a number has been increased, but we do not have an infrastructure. Only major institute, Apex Institute, they're maintaining. Like your institute, are maintaining both smaller and higher animal. But if you go to peripheral college, you'll hardly see that they do not have a facilities for that. That is what the problem is.
Thank you, sir.
Thank you.
Thank you for answering the query, sir. Sir, as you mentioned, unless there is a proper alternative to animal studies, we may have to resort to animal studies only. So thank you, sir, for such an informative session.
Thank you. Thank you very much.
Our next speaker for the day, Dr. R.K. Dikshit, Professor at the Department of Pharmacology and Therapeutics, KGMU Lucknow, is a stalwart in the field of medical research in India, having published more than 166 publications. Sir has also previously held many distinguished positions, such as the Principal, Autonomous State Medical College, Ghazipur, Uttar Pradesh. Due to an emergency commitment, Dr. Dikshit could not be with us, but as the true professional that he is, he has sent us the video of his session, which we will now be playing for you.
A very good afternoon, everyone. I am thankful and fortunate that Amrita Institute of Medical Sciences and Research Center has invited me to share my thoughts related to the alternatives to the animal studies. I am especially thankful to Dr. Princey. Now I'll start my session with some history of the animal experimentation. Animal experimentation, where and why they are required and necessary as of today. Laws and regulations related to the animal experimentations. Alternatives to the animal experimentation, there are two formulas. One formula you must all have heard, R formula, that is that I'll tell. The S formula I also found, and that I will share. In vitro methods, in silico methods, advantages and disadvantages of alternatives to the animal experiments. And then I have prepared my own formula, that is ABCDEF, for the alternatives to the animal experiments. And last, the summary. If you see the literature, it is full of controversies related to the animal experiments. And some are against the animal experiment, some are in favor of an animal experiment, and that actually controversies was actually became more when the COVID came. The COVID actually showed that all the alternatives of the animals, all animal experiments cannot be replaced. And we can never think of any vaccine which saved our life, that was against the corona, and that actually was because of the help of the animals. So though animal experiments, they are to be reduced, but cannot be replaced 100%. And as of the PG teaching is concerned, and our most reputed Dr. Ravinder sir has given a wonderful editorial that suggests that the postgraduate teaching, this little bit of exposure or relevant exposure to the animal experimentation is must. Whether the students will go to the industry or they will remain in the academics, but the real animal experimentations in the postgraduate training as of now is must. Every time, rather most of the time, not possible then, do experiments on those who are closer to the. So once any compound is being synthesized or found out, we are unknown about the action of that compound over the living body. Then we have to go with the living which are actually lower at the hierarchy. And every time you cannot go just to the person itself or the human being. Ideally, it should be in the same human to whom it is going to be applied, but this is ideal, and ideal doesn't exist. Then we go with the animal closer to the human in biochemistry, anatomy, and physiology. Then in vitro cell culture, and then animal away from the human, like lower in the stature. Then we have in silico computer generated predictions and all this, and some of the other methods. So I'll go one by one. If you see, the animals are in between. We start with the abiogenesis, then came the prokaryote, and ultimately the animals are in between, and we are at the top. But when it comes to the experimentation, they become the first choice. The reason is they are very low in the hierarchy, and most of them, they don't give the actual representation of the human body. So that's how, and here we are very close to, we cannot go with the experimentation over the apes and all, and because of that, the experiments on the apes and the higher species has been banned in most of the countries. So these becomes the first choice. If you go with the history of the animal experimentation, it is very old, and as far as you can search about the history, that was about the Aristotle, and he also experimented on the animals. Maybe it was surgery, it was medicinal use, or anything. Then very reputed scientist from the Ibn Johar, he also was known for the animal experimentation. And our father of physiology, Claude Bernard, he was known for having vivisection of many animals, and that actually led to the immense increase in the medical knowledge, safety of the pharmaceutical, cosmetics, and chemicals. So animals are used for not only for teaching purposes. Teaching is the one other, because most of the animal activists, they actually are against the teaching, the animal use in the teaching purposes. And if you see, this is at the lower part. Most of the time we get improved understanding of the biology, we have a improved understanding of the pathology, then research development, including the drug development, health and medical research, toxic potential, cosmetics, ecological research, and the education becomes at the lower part. So we actually utilize less than 3% of all the animal experimentation during the education, to whom we are concerned. And these, if you can replace, they can be replaced totally. But ultimately, here the drug development and all, there some share of the animal experimentation will be required even in near future also. But though the proportion can be reduced. So if you see, the education is very less, and out of which the pharmacology, we utilize lesser proportion of the animals than others which are used in toxicology, oncology, genetic modification, immunology, and all this. So animal used in the experiments are mostly, they are rodents, fish, and if you remember, the mice, rat, fish, and birds, they actually are the most common. And how to remember for PGs, the mice, rat, and fishes, that is MRF bond brand, that you remember. The mice, rat, and fishes, they are actually the most common, because the examiner will ask which are the animals which you use, or which are used most commonly for the animal experimentation. That is mice, rat, followed by the fishes, and followed by the birds. There are many tricky questions which may be related to the philosophy, may be practical, and humans are unique amongst all, and we have to actually go with the moral decision and philosophical decision, and to be also practical. And that balance should be their practicality as along with the philosophical and morality. Animals are certainly not perfect model for the human. That is one thing which actually gives against the animal experimentation, because most of the diseases, most of the time, the drug was actually investigated in the animals and found out that it was not having a replication in the human as such. So that is against against the animal experimentation. Second is the animal anatomy biochemistry is not 100% mixed, and that is true. But on the other hand, if you don't go with the animal studies and all, then we can have a regulatory issues for the drug development, cosmetics, and all the other things. So different views are there, many oppose, many support, and most promote for the alternative. That is the best thing. So alternatives are being appreciated by all, and at present, but unfortunately at present, the guidelines are underdeveloped. Reasons in favor of use of animals in biomedical research. The most important is nothing can substitute the complex function of the living. Living are having a very complex mechanism. They are highly complex mini computers, or the mega computers, and that functions are actually, they cannot be replicated by any of these supercomputers. So that is one thing. Second is most of the DNA, our DNA, that can match with the some of the animals like mice, though it is entirely different in morphology, anatomy, and all other things, but the DNA is 98% same with us, and that actually gives in favor of the research in the animals. Similarity related to the diseases, some of the diseases which are actually present in the animal, they actually replicate similarly in the human also. Shorter half-life, you can study the effect of the drug throughout the life, and that actually can give a clue that how the drug will behave in the animal human beings. Projected impact, removing the animal completely from the lab. If you remove all the animal from the lab, then what will happen? Now this may give the negative impact over the development of the new and vital treatment for the human as well as the animals. And if we are over dependent over the technology, sometimes the havoc may occur. And naturally, the ecological impact and regulatory issues are also there if you remove all and each and every animal from the lab. But there is a need for alternative, because every time we cannot go with the sacrifice of the animals. So there are ethical issues, and because the ethical issues, unethical handling of the animals leading to the damage of the psychological stress, there is a issue of pain, requirement of the skilled manpower, consumption, protocol, high cost, and the last one is that lack of the guarantee of the translation, what we get in the animal into the actual humans scenario. So we have to be little bit of rational approach, and that's how we have to go. That we can be a little bit in the middle. That whenever not possible, then we utilize the alternative. Whenever it is not possible to go with the any other mean, then we have to go with the animals only. So 3R everybody knows, and that principle was way away back given by the Russell and Birch in 1959. The first R is reduce, second is refine and replace. What you reduce? Reduce the number. How you can reduce? By reducing the replication, by reducing the duplication, by applying the proper statist during the time of framing the hypothesis and the methodology. So that's how we can reduce. How we can refine? We can refine by reducing the pain, by giving the proper anesthesia, by giving a proper infrastructure for the habitat and all these food and other things. So that they suffer minimally. And how we can replace? And we can replace animal with some of the computer designs, in vitro, cell lines, in vivo, in silico designing and all these things. So that will replace the animal. But 100% replacement, I am again saying that it is not possible. So we have to go by these three, that is reduce, refine and replace. So now there are, if you see here, reduction, replacement, refinement, in vitro, in silico, and all these I already told. Even the human skin irritancy test, that will replace the unnecessary use of the rabbit skin and all other for the irritancy test. Then human blood can be utilized for the pyrogenicity, which was actually utilized in the rabbit. So microdosing is still in the human volunteer. They can be a substitute for the development of the alternate to the animal experiment. So that is replacement. I have already told, the preferred use of the non-animal methods over the animal methods whenever it is possible to achieve the same scientific. The first thing is, if it is achieving the same scientific aim with the all appropriate accuracy, precision and all these, then we replace, otherwise not. So that is the first thing. In vitro experiment, cell cycle, tissue, organoid, taxonomes. They are most of the labs, they are actually working on this, and these are available not only in the developed countries but in our country also. So in vitro experiments are very well designed, and they are giving a good results and replacing the need of the living animal. Then QSAR studies, they actually give the lead related to the properties of the drug. Maximum use of the bioinformatics, because the era is of computer and all this biology with informatic technology, that is giving the boon, and that has given immense advancement in the science. So the most important, you can have lab on chip. Everything comes in chip. So if you remember your old days, we were having a floppy, and then came the disk. Then now it is a chip. So everything is in chip. So that's how the lab is also on the chip. Maximum use of the computer-assisted learning, which is actually utilized for the UG, PG, pharmacy students and others. So that is one character which will actually reduce the use of the animals in the educational purpose. So that is the very important thing. The CAL is very important, and this will actually reduce the use of the animals or the requirement of the animals for the educational purpose. Use living If the living is required, then actually we can go with the lower strata. And here we are fortunate to have a very important fly, that is fruit fly, Drosophila, and that actually gives very similar results with the human being. Then human skin itself, that can be utilized. Human blood itself can be utilized for the pyrogen testing and all these things. Then human itself, by administering the microdosing, that can be utilized for the use. The reduction is a method that enables the researchers to obtain the comparable levels of the information from the fewer animals. So that I have told that reduction can be done by strong hypothesis, statistical calculation and analysis, having imaging technique, good imaging technique, sharing the data and reducing the duplication of the experiments. Refinement can be done by better housing, better and gentle handling of the animals, then imaging, reduction of the pain and better pain relief, shorter experiment. We can go with the shorter experiment, and we can go with the better anesthesia, and if needed, to kill the animal with a high quality euthanasia, so that the minimal suffering occurs to the animals. So conventional testing was like this. Here we think that it will be there, but 100% it will be there. Everybody doubts. So if you go with the alternative, the animal started with the 1980s up to the 2015. It started with the purified enzymes. That was in vitro studies. Then 2D cell tissue recombinant DNA enzyme came. Then 3D cells tissue. Here this was the very important. This was the era when actually scientists thought that okay, the cells can replace the whole animal, and that cells tissue or the bunch of the cells, they can actually replace the whole live animal. Then came the chips, and the chips are the microfluidic chips that they are being developed, and they actually mimic with the organ. They actually give the relatively closer result to the living being than the cell itself. So here this is a type of transition from living to the non-living with the result similar to the animals. So it's all about in. So what is that in? The in vivo, in vitro, in situ, in vitro, in silico, in chemico, all this. So it is everything about the in. So we started with the live animal, then came the in vivo, in vitro, in silico, and in chemico. But mind it, live is live, and nothing can replace the life. So if you see here in silico computational modeling, in vivo modeling, in vitro organ modeling, so all these, they are being tried and have given comparable results also. So I'll go with the little bit details of the each and every one. The cell culture and tissue engineering is there, and that is utilized for the development of the monoclonal antibodies and the cosmetic products. Cell culture and tissue engineering, but here one should think that the nourishment of this is actually by the live only, and because of for that the many of the fetal women, fetal cows have been sacrificed. So again the cruelty comes in between, though it looks without live, without involvement of the animal, but at the background the animals are being sacrificed. So human skin equivalent test that can be, and there are many models like epiderm, episkin. They have been for irritancy. Fungal model, they are red mold. They are a good model for circadian rhythm. Then prokaryotes like E. coli and Bacillus subtilis, they are utilized for genetic and molecular studies. Then term is organoid. Organoid is a 3D model of culture cells which actually give almost similar shape to that of the organ, and they can be derived from the embryonic, adult and induced pluripotent stem cells. So organoids are very promising, but they are not perfect. So don't think that they are perfect. Then just like organoid, the tumorides are there, that is the cells from the biopsy from the cancer patient, and they have actually given the results as if the person is having tumor itself. So human toxome, that gives the toxicity studies in vitro testing. So in vitro invertebrate Drosophila melanogaster, that is actually zebrafish, that is at a lower strata, that can be utilized for the as a replacement of the animals, bigger animals. Advantages of using zebrafish are multiple, right from the short reproductive life to the higher spring and all other things. Then zebrafish in research can be utilized in toxicology, in pharmacodynamic and kinetic studies, cancer research, genomics, economics, ecotoxicogen, and other things. Then comes the era of the computer simulation, and here we can have a high throughput screening for the as a replacement of the animals, and this is most advanced upcoming advancement in the alternative of the animal experiment, that is microfluidic chips or the cell chips. These are very important, and they actually give almost similar results related to the live tissue. So microfluidic chips are cell biochips. They are perform the perfusion culture, and then a whole organ is on the chip, that is actually mimic, and then they say that whole lab on the chip. So everything is on the chip, and that you just put a drop of the fluid and everything is channelized, and that gives the similar type of results which a live human being is giving. So these are the terms which are actually being utilized. So microdosing can be utilized as a replacement for them, but microdosing can be a cause for the false negative and the false positive test also. Then there are certain research initiatives globally. They are Sudet 1 and Europe Ecotox. These are two initiatives which are actually replacing more and more animal experiments. Then there is one site that is on Norikopa. Norikopa is a Norwegian national consensus platform for the advancement of the 3R, and that actually has many models, more than 300,000 audiovisual ads, which actually give the learning and educational training for different types of and our Pharma, the Pharma is under that also. So then other are Brain Tree Scientific Incorporation, then Sealed Program, then Digital Frog, Alternate. There are many. So you go with the search over the Google and you can find out many. But I'll focus over the two. One is Experimental Pharmacology series developed in our India only under the able guidance of the Ravindra sir. And X Pharma Pro gives a very relevant educational material relevant to the PGs and undergraduate pharmacy students and all those who are actually involved in the pharmacology training. That can be actually taken from the GEM portal. The second important upcoming platform is Medimation, and they are also having a very nice type of educational videos and other educational material which can be utilized for the teaching and training of the. So Medimation is a platform which actually not only for the pharmacologists, it is for the X Pharma was for the pharmacology only. Now Medimation is for not only the pharmacology, but it is for physiology and the other things, pathology, So they are having a short videos, then animations, then other educational material which can be utilized for the teaching and learning purposes of the UGs, PGs, and of course to train ourselves, that is teachers. So these are some of the short films which can be downloaded, and they gives the actual scenario just like you are seeing in the live demonstration. So they are excellent one, and they can be utilized whenever one needs. That is how the CAL can be utilized. So human patient simulators are there, and they are for good for the hands-on training over the human torso. Human volunteers can be utilized, and that microdosing already told. Then population research, you go rather than inducing anything over the animals and going into the intervention, we just go to the population and we see how the population reacts with the particular toxins which have already been released and has been exposed. Role of OECD is very important in promotion of the alternative, because if we share the quality data with each and every country, then naturally the number of the experiments will go down. Everyone has their own opinion, so there is nothing like one black and white. So use of animal can be because of the regulatory issues, because the drug cannot come in the market, or you cannot go directly to the human being because of some regulatory issues. So you have to go with the help of the animal experiments. Lack of validated alternatives and risk of litigation. So use of alternatives, they may be because the in vitro methods are cheaper, though the first initial cost is very high, but once the recurrent cost becomes very cheap, then they are very fast, and the reproducibility of these alternatives are good. So why and when animals should be used? Number one, when there is no other way to do the science. Number one, this is a law, and law in UK, European Union and all other things, that if you don't have any other way, then you have to go with the animal. So only when the sound scientific justification of the use of the animals, due consideration of the R and S. So I'll tell what is S. So S is actually that is science with sensible knowledge of the science and sensitivity to the emotions of the animals. Is it possible to replace animal experimentation completely? It is big no. So take home message: all concepts and comprise all replace, refine, reduce, revise, reduce, and reuse and rehabilitate and register. Just like the human clinical trial, the animal registry should also be there. And all essential principles of the human experimentation should underpin the training the personnel. So thank you very much, each and everyone. Any suggestion, any question, comment, you can post in any of these platforms, or just you can WhatsApp on me on this number. Thanks a lot, each and everyone. Thanks once again.
Thank you, sir, for making us realize our unique moral position where we must pose the question at what cost. There's an important announcement. Kindly fill the Google form that has been put in the chat box so as to receive your certificates, and and please fill your name as you wish on the certificate.
Let us now dive into the controversy on animal ethics. We are all probably in a dilemma regarding the use of animals in education, training, and research. To further elaborate on this issue, I would like to welcome Dr. Ajay Prakash to this virtual platform. Sir is Assistant Professor at Department of Pharmacology, PGIMR, Chandigarh, and Deputy Coordinator, Pharmacovigilance Center. Sir's interest in non-clinical, preclinical, and clinical pharmacology has led him to publish more than 95 peer-reviewed articles in national and international journals, as well as 3 books and more than 33 chapters in international publications. Over to you, sir.
Yeah, thank you so much. I'm audible?
Yes, sir.
Yes, sir.
Yeah, before going to my presentation, if anyone have question regarding the last presentation, then I can answer, or the question answer will be taken after my presentation. Okay. Thank you so much. So what I'm going to deal with today is that the animal ethics. You all are aware that yes, there is lots of the discussion on that, the use of the animals, and whether to use the animal or not, whether to use the animal in the PG curriculum or even in the PG curriculum, or during the research or not. So there are so many questions, and even the you can say the debate is also there in so last 1 or 2 decades regarding the use of the animal. And after that, as the Professor Medhi already told, that there are so many, all the MCI, PCI, UGC and other universities in India have also given the advisory for use of the animal during the teaching process. Okay, so see, the main thing involved in this during the huge and cry regarding the debate of the animal is only to that whether we are using the animal ethically or not. Okay, so most prominent thing is that if we are using our animal judiciously, or if we use our animal where we need to use the animal, so I think there is no problem at all. So just deal with that. First, I think you all aware about the PETA, that is the People for the Ethical Treatment of Animals. So just start with this because they have raised so many voices regarding the ethical concern of this animal experimentation during the education and research. So they have three main motto. That is the animal experiments are whether they are wasteful or unreliable, whether we can rely on the result of those animal experimentation for the research purpose or not. So it is the questionable for them. Second is oversight and regulation, whether the whatever the regulation present in India or even abroad in all the cities, all the countries, whether it is whether the rules and regulation and SOP, you can say the standard operating procedure of all these are up to mark to see the ethical concern of those animal ethics are good enough or not. Okay. Third is yes, well taken and by Professor Dixit sir, and alternate to the animal testing. See, whenever we are dealing with some experimentation, we cannot first hand we can say that yes, the result will be come from alternative of any alternative to the animal experimentation or from the animals. Yes, if there is some test is available or you can rely on the result of those alternative methods, yes, you can use that. But if in vivo experimentation can be done by putting the all the efforts, all the efforts means if you use the animal ethically as well as per your protocol, what the Institute Animal Ethics Committee have given you for approval, so it will be, I think, done most of the things. So just in data, just copied from the PETA site only. Why they are saying like that? Animal experiment are wasteful. Reason being, in it is a data of 2019 that almost 1 million animal were held captive laboratory and about 100 million mice and rats were was killed or sacrificed during the experimentation. Canada, yes, 2020 data says that 5.07 million experiments used in the experiments, 543 animals subjected to severe pain near or the above the pain tolerance threshold, or they have used the less anesthetized anesthetics agents. Okay, it is the data of the Canada. Third is the United Kingdom data that shows that 2.88 million procedures were done on the animals and 1.4 million experiments were done. So what is happening? What is happening in India? So if I search for that data, but I am unfortunate to get the data exactly. Even the data also have not given these data. However, what is our concern is that our concern is only this that it is also come to the that nature publication what the Professor Medhi was taking you through, that from last so many decades, so many billions of the animals was used in the Indian context, and there was no single NCE was discovered in India. Means that we have used these animals uh, means on trial basis. Simply, you can say on the trial basis, but we do not have any opportunity to get the new chemical entity in the market. So this is the main problem in India. What is our thought is here. So what is thing is that if we are using the animal daily in the experimental purpose, even in the research purpose, even in the pharmacokinetic study of the regulatory study, even we are doing the toxicity studies for the regulatory studies. So why not the new chemical entity can be delivered or discovered in India? This is the main limitation PETA, what the aspect of the PETA is the different, but our aspect as a academic section or as a regulatory aspect is different. If we are using so many animals, but we are not able to discover a single NC, so what is the use of using those animals? So better we can use the alternative to the animal experimentation, as the Professor Dixit said. But see, we have both the points, means whether to use, whether to not use. See, stopping complete animal is never be advised and never be successful for the drug development process. So question is that why we are using animals or why we need animals single. So simply we say, if we talk about these scenario, the two aspect of the drug discovery process means vital, the animal is really plays very important role in the drug discovery. As recent example, we have seen that during the COVID-19 vaccination discovery, lots 90% data is generated from the hamster or mice or rat. Means without that we cannot come to the conclusion that yes, these product is going to be used or will be safe in the humans. So it is not the thing that we can stop the animal experimentation. We have to do the animal experimentation. What is the criteria of the drug discovery is that when we are getting this target selection over there, then lead identification we do. We go to combinatorial chemistry, mean in silico. That is also one type of alternative medicine where we do the in silico drug docking studies, all those. Where we do the in vitro study, that is high throughput screening. So this is a process. We cannot say that one point of doing this is wrong for the society or wrong for the animal is not. It is the need to do, because this is the step that we have done on the computer simulation. Yes, computer simulation is good enough. Whether it is going to work on cells or not, we are going to do for the primary cells. Whether it is going to do for the cells to the tissue, whether it is active on the tissue or not. So we are going to 2D culture or 3D culture. So if after doing that, whether these drug is going to work in vivo condition or not, because whatever the condition we are making in vitro is totally artificial or totally what we proposed, that is also consist of several errors. कभी CO2 काम नहीं कर रहा, कभी O2 का composition ratio is not good, okay, कभी media is not better. So limitation is so many. But if you talk about the human as a human or even one mice, the internal environment of a mice, internal environment of the rat, you should not check. You cannot able to change. Understood? So whenever we are doing this drug discovery process, we always look for the whether how the drug will behave in the body simply. So this is, if we see the typically we are genetically also that it genome by 90 to 99% mice and these are same, marmosets are the same. Okay, so this is the need of doing the experimentation of in the in vivo condition. So this is one type of the process. Before that, how we can go to the toxicological studies and how we can go to the first in human dose calculation. So these are the some point we need to be debatable and we need to be explained properly. So overall, when we talk about the ethical concern, understood that we have two aspects of seeing things, that is the academic level and one is the regulatory level. Academic level is nothing but what we are doing in our MD level, we are doing in MPharm level, in PhD level, means we want to do some project or to partial fulfillment of our degrees, means PhD kar lo ya MD kar lo, MPharm kar lo. So this is the for the ethics arena is that you have to take permission from animal ethics committee. It is based on some rules and regulation. Okay, but when you talk about the regulatory, yes, as Professor Medhi said, as Professor Dixit said, you have to go through the OECD GLP guideline. That is very strict, very stringent. You have to make a count of each and every one, step by step responsibility you have. So each, see, the seeing both ethical concern from the academic level and regulatory entirely different. In academic zone, you are just doing your thesis, you are submitting your thesis, and you are going to publish some article, or if it is innovate enough, you may and may not go to for the new chemical entity. But regulatory is always be concerned with the clinical study, always be concerned with the your toxicity study, side effects study. Okay, so the perspective of ethical concern with these two are very, very different. So overall, the difference is that why we are doing these things, because we need to know that yes, phylogenetically the level of these animals are different. As the Professor Dixit have shows the trees, phylogenetic trees of all the animals. So the briefly, if we talk about the ethical concern or animal ethics approval, so it was not from 2020. Before, it was not there in the zebrafish. But after 2020, CPCSA have issued separate guidelines for birds, separate guideline for zebrafish, separate guideline for higher animals. But before that, it was not there. So what happened if we say ki, animal ethics will apply after the phylogeneticity of the fish and birds? Means fish पे हो सकता है, birds पे हो सकता है, mice, rodents पे सारे पे हो सकता है, monkey पे हो सकता है. Understood? So these are the scenario where we have to talk about the ethics. Ethics means, what do you mean by ethics then? What do you mean by ethics? Ethics already have told. We have the 3 R's. This is the major principle. I think all of you know, and already Dixit sir have told that in detail. The what is the reduction, what is the refinement, what is the replacement of these 3 R's. But problem in that, even after the, I think it is came in 66. So now after 50 years after that also, these principles are not followed well. Why it is so? Because we have now 5 R's: reduction, refinement, replacement, rehabilitation, which is recently last year, I think 2019, CPCC have included rehabilitation as a 4th R in their guideline. Okay, and 5th R, that is the responsibility, is we have enrolled in our book. So responsibility, why responsibility important? I've just emphasized because all 4 R is there, you can go and read. But why responsibility is 5th R? Because if you are not found yourself, or experimenter, or whatever the researcher is there who are working on the some protocol, found themselves responsible for all these 4, all 4 are worthless. Understood? So if you are found yourself responsible enough to work for these 4 R's, that is the reduction, refinement, replacement, or rehabilitation, then only the you can say soul of these 3 R's will be sustained and protected. So whenever we talk about the animal ethics, always remember, in India, we are not going anywhere. Only we have the committee for the purpose of control and supervision of experiments of animal. कभी कभी ये PGO पूछते हैं what is the full form of the CPCSA. Always remember, this comes under the Ministry of Environment, Forest and Climate Change, and now it comes under the chemical and fertilizer. Okay. But what is the basis of making these guidelines? The making of these guidelines is based on the Prevention of Cruelty to the Animal Act 1960. This is the main crux which you have to remember for how the CPCA generated the guideline. This is just based on these. They have contained 2. One is the forest animal, second is the experimental animal. But we are dealing with the experimental animal, so we are going forward talking about the experimental animal. What ethics says, what the CPCA guideline says to you, that whenever you are going to use your animals, or when you are going to use your any animal experimentation, the care and handling should be humane. Means that should not be in pain, that should not be in some discomfort level. So nowadays, even in housing condition also is changed severally. जैसे हमारे we have in kindergarten होता है ना, जो small babies have some toys to play. So now animal are also kept like this, that if you have animal like you have the animal houses and housing of these, you want to improve, you have to give small toys like play to the animals, means for their well-being, to improve the health and well-being. So good laboratory practice, again I am saying this is only requirement for the regulatory study. For the thesis purpose, it is not a requirement, but yes, if you follow the SOPs for your day-to-day practice, it will already advisable, but it is difficult. So promote the human care of the animal. So over the crux of the CPC guideline is that you have to take care of your animal, whatever the issued to you for the experiment. Second, whatever the experiment you are going to perform, that should be the painless, or you can reduce the pain, or whatever the procedure you are going to do for the experimental purpose, that should be humane and there should be no pain. This is the main crux of the CPCA or any ethical guideline for the animals. So overall objective, if you say, provide a specification that will enhance the animal well-being quality in the pursuance of the advancement of the biological knowledge that is relevant to the human and animals. So nowadays, if we talk about the CPCA, they have further constituted their own ethical committees in the institution level. That is known as issued animal ethics committee, and second is the issued biosafety level or biosafety committee. Okay, so these two institutional level committee who work for the CPCA to maintain the animal ethical guideline during the experimentation in drug discovery process or any. So these are the guidelines which are come in the 2020, which were not before or not come under the arena of the ethical permission. You can say if you want to do the experiment of any birds, poultry birds, or any birds, or even you want to do the animal of the large animal. So these guidelines or use or rehabilitation of those animals, these guideline was not there. So these guideline was given to the 2020. Again, remember that this is the guideline for the reuse and rehabilitation of large animals. It is not for the use. For the use of large animal is already there in the experimental animal section. But if you want to rehabilitate this, you want to reuse this. So what are the criteria? You go, it all are the free. You can go and CPCA guideline, CPCA website, and you can download it. The recent one last year came that the guideline for the CPCA for the experimentation of feces. It is now, previously it was not there. Previously the zebra feces even was exempted from the ethical arena, but now after 2020 issued of these guidelines, CPCA guideline, now feces are again, you have to take the permission for doing zebra feces experimentation. Previously it was found that for the day 3 or up to day 5 experimentation was allowed for the zebra feces. After 5 days it found to be adult, and there was, you need to apply to the IEC, but now from the first day you have to take permission for the feces also. So these are the recent guideline for the CPCSA. So overall, what is Animal Ethics Committee? Animal Ethics Committee is a member of 6 to 7 members simply, but mandatorily 5 members should be presented at that meeting in where the meeting is going to conduct. So who are those 5? Among those, or you can say the who are these 6-7 members? One is the biological scientists, 2 scientists from different biological discipline, one is veterinarian, and one is scientist charge of the animal facility, and scientists from outside institute for the unbiasedness opinion, and one is non-scientific social aware member responsible for the CPCA. Means some CPCA nominee should be there, from the CPCA who trained nominee is there. So they should be a principal or important part, taking important part in those meetings. So total 5 members should be there. One is the layperson, or you can say the social worker, so that they can check the social one type of those social responsibility of doing these experiments. Yes, beneficial for the society or not. So these are the rules. So what is role? Role of the IEC is only to approve the experimental protocol which is applied to them only to the small level. Small level means rodents. If you want to use your large animals, mean marmosets, or you can use the pig, or you want to use the beagle dogs, then the IAEC know that comes under directly to the CPCA, not under the IAEC. So whenever you want to do in the large animal experimentation, directly you have to apply to the CPCA through IAEC. Means you have to apply to IEC, but you have to told that I want to do on the large animals, so they will send your application directly to the CPC. So, but channelizing should be there. Through it will go to the IAC only, because CPCA nominee is there. They will decide yes, the protocol is scientifically, ethically sound enough to send to the CPCA. Okay. So second is the issue institutional biosafety committee. In these, who are the concern which comes under this is that the mainly the chemical who are hazardous in nature, who have the radioligand or who have the DNA modifying capacity of mutation जैसा कुछ हो सकता है, तो these type of the protocol containing those type of the substance should come to the biosafety committee first. The sequence is the applying for the animal ethical clearance is that if you are using those type of the ingredients or drugs in your protocol, then first you have to take the approval IBC or IBSC. Then after the approval of issued biosafety committee, you have to apply to the IAEC for the final approval for your experimentation. So the member is head of the institution or his nominee, or you can say the chairman. 3 or more scientists engaged in the DNA work or molecular biology. He have formed enough concept of that alteration, mutation, geneticity. Okay. So 1 member from medical qualification, biosafety officer may be there. 1 member from nominated by the Department of Biotechnology, means DVT say 1 member will be there. So main role of doing IBC is that they will give clearance to the proposal which are falling under the restricted category, training, personal or biosafety, means biosafety 1, 2, 3. Okay. So we are going to take the approval from this. So adopting the emergency approval also, wherever it is necessary, we have to take the approval, then approach to the IEC. Okay, so these are the guidelines. I'm not going to tell. I think it is a freely available CPCA guideline. You go and type on the Google CPCA animal experimentation guideline. First option you will get, you can download and go through. But what I'm given in the highlights is that if you see each and every headings or subheading, that is adequate veterinary care, animal procurement, quarantine, stabilization, surveillance, diagnosis. So they are taking care of each and every health care aspect of the laboratories or animals. Okay. But the animal care of the each aspect means personal hygiene should be there, animal experimentation, what the hazardous agent involved, that list should be there, surgical procedure, duration experiment, means each and every care what as much as you can take care of the animal that is there. Even the condition of environment also should be stringently having the each and every SOPs for temperature, humidity, ventilation, power lighting, noise free, because what happening mainly I just like to emphasize on the power and lighting. Whenever we want to do the experimentation on animal, for them the day is the night, and for night they are, because they are rodents are the nocturnal mainly. So when we are maintaining the light and dark cycle, if you go to the many research paper, you will find 12-hour to 12-hour light day cycle. It is opposite to them, means complete night day for them, because they are going to kept in the high light. After that, in morning light is off, means now it is day for them, so that you can do the experimentation. See, whenever these cycle is also altered, your result may vary if it is related to CNS experimentation or some. So animal husbandry, caging, all those. So there are so many. Each and every aspect is there. Each and every you can say biological parameter, each and every hematological parameter is given there in the CPCC guidelines. Food, water, bedding, standard operating procedure, personal training. So each and every, and finally euthanasia. So you go and read. I'm not going to detail. So important thing that when you are going to apply for the IAC approval, remember that we are come across with 2 major forms. First form is Form B, which is for your submission for the IAC approval. That is known as proposal to submit for your protocol to the IAC. And second is the Form D. Form D means when you got approval from IAC, now you want to issue your animals, then you are going to fill the Form D for the required animal requirements. What the number of animal you want to require, you are going to fill Form D. These all forms under the CPCSA guideline. Form C is not for the experimental, it is only for the establishment or the facility who are maintaining those. So they have to take record of each and every strain, each of every species, each and every gender wise, male, female. Even outside it is also maintained age wise, I mean who are younger, older, and what is the last 3 to 4 history of their research. Means the point of research done already on them is also kept into the under these forms. So these are very, very important as per the animal ethical concern is there. So that you will get proper idea that the animal you are going to use is gone through how many experiments or whether it is new or naive animals. Understood? So it all have the impact in the research. So this is the form. We have two things, section 1, section 2. So registration, section 1 only contain one page, that having the registration from the CPC, what is their number, for how many years they have registered. So this is the brief I feel you can get from your institute ethics committee. Then second section is complete about your protocol, your procedure, if any surgical procedure there, you have to explain properly. If some rationale of those, why you want to use. Now see why we are using alternative, because they have already specified into the animal ethics committee form that rationale for the animal use. Understood? Rationale for the animal use means you have to define well whether you want to use the animal or not, or why you want to use the animal in your experimentation. Okay, they need each and every aspect of procedure, whichever the procedure you are going to do, whether it is surgical procedure, or if you are doing the surgical procedure, what are the post-surgical planning you have done to reduce their pain, reduce their infections, reduce their well-being, or to keep their well-being. Okay, if any hazardous agent is there, you have to go to the IBC form, and after IBC form approval only you have to apply to the IEC. So now coming to the second very important thing, that is arrive guideline. I think during Vikash sir lecture there was some question also for the arrive guideline. See, arrive guideline is nothing but it is not related to ethics, but it protect ethics. Understand? Arrive guideline is the guideline which give you the guideline to make up the protocol in such a way that ethical soul of ethics should maintain. Now, you see that in clinical study, we have ICH guidelines, we have GCP guidelines, there are several guidelines. Why these guidelines? Because we need homogeneous data all over the world. But in preclinical study or in vivo study, we do not have such guideline which follow some guideline and give the homogeneous sample. So right now in 2010, arrive guideline came. Now it is arrive guideline 2.0, means it is somewhat modified from the previously published arrive guideline. So they are mainly concerned about a study design, sample size, inclusion exclusion criteria, randomization, blinding, outcome measure, statistical measure, experimental. See, means they want to make your protocol in such a way that all the protocol have some information regarding the which type of the animal you are going to do, what is the number of animal, as we are doing the patient number, what is the design of clinical studies. So the same principle arrive guideline is there, so that whatever the data we are getting, that may be repeatability and applicability to the other centers also or other countries also. So overall, the idea of doing this or for this arrive guideline is that improving the reproducibility and reliability of the findings and repeat the build up the work for the other researcher, mean producibility and reliability of the result. So arrived guideline is not directly involved in the ethics, but arrived guideline very much involved in ethics to keep their soul maintained, so that whenever you proposing some study design, proposing some sample size, you should keep into mind the 3 R's: reduction, refinement, and replacement. Whether if there's a need, you can reduce your sample size from 10 animal to 6 animal or 5 animal, like that, if it is feasible. So that depend on the scientific output, objective, very subjective. So I'm not going to detail. So this is also free. You can go and download this and study this. So if any question, you can mail us, we will clear you. So these are the main 5 to 10 point. That is the point to be remembered, take home message for that, that whenever we want to do the experimentation, understand that we want to use these with the respect, and animal dignity should be maintained. Responsibility of considering option. If you have the replacement option or having the result outcome is same as the in vitro parameter, better to change to the in vitro parameter, so that it will also exempt from the IEC approval. The principle of proportionality and responsibility is also there. That is the responsibility for the considering reduction of that is the reduction of is also there. Refinement is also there, and responsible maintain the biological diversity. Biological diversity is that we always say that whenever you are going to develop some animal model, suppose you want to develop for the diabetes, suppose you want to do for the epilepsy, you want to do the Alzheimer disease, always you go for the sensitive species to, or select the sensitive species to develop, so that it is not exactly related to the humans, but the resemblance is there. Genome wise, 99% genome wise is same. So resemblance is there. So whatever the data you are going to get, it is having the more and more plus minus HD is always there, but it have the some translation value. Okay. So habitat, that is habitat is there. Responsible for openness and sharing the data. If it is reproducible, so you can share the data. Expertise or animal, it is very, very important. It is not necessary that if you are going to use the animal, so if we hold the teaching of animal experimentation in PG level, how we can go to produce more and more expertise people for the researcher who we can say the researcher in future. So this is very, very important and necessary also. So what I can say, as if clinically, we are always discussing about the PICOT formula of the protocol. That's a PICOT. Okay, that is very, very important to follow the soul of a protocol to have a reproducible data. We have SIP code for the research. We have recently decided, and this is SIP code for the in vivo study. What is SIP code? You have to first select the very specific or ideal model for your disease condition. So species is the first. Second, in the indication, for which indication you are going to use, accordingly, whether you want to go for the acute model, whether you are going to subacute model, where you are going to chronic model, you have to, as per indication of those in the clinical section, you have to decide indication for this. Means treatment duration, you can say. Okay, so P is nothing but the procedure. In P, in the PICOT form is the population, but here it is the procedure. It is very, very important in the animal experimentation, which procedure you are going to follow. Where you are going to inject IP, you are going to give IV, or whether you are going to give the oral treatment of the intervention, or you are going to do some surgical procedure. So ethics changes in each section. So procedure is very, very important. C is the control. Already Professor Medhi have told that we have to follow the specific study design. So if each and every section, we must have control, positive control, negative control, naive control, so that you have firm idea of the outcome. Okay, so always outcome endpoint is the main for any type of the experiment, you can say. So outcome and endpoints for the O, and T is nothing but the time and duration. for what time you are going to use or for what time you are going to give the intervention and for at what time you are going to do the surgery. It is very important, and for the pre-operative care, post-operative care. So SIPCOT is the main thing that you have to do for better understanding of the protocol, better suiting to animal ethics and approval for from the animal issued animal ethics committee. So just remember SIPCOT. So overall update is that you all think, we have restricted use of these PG due to the 3 Rs, but 3R never says to stop those in the curriculum, so that we have to generate more trained animal and more trained the researcher for the animal experimentation. So if we are going to stop, how we are going to do the or produce the expertise, I don't know. So it need debate scientifically, altogether complete scientifically. We need debate on this. So once again, thank you so much. I think I'm on the time. So once again, if any question related to this previous, I'm here to answer.
Are there any ethical consideration regarding invertebrate use in experimental studies? Question is asked by Hemavati.
No, nowadays there is no such guideline. But yes, but if you go slightly above than fish, you have all the ethical guideline. But invertebrates can be available.
So Vijita has asked, is it necessary to make IBC if there are no animals available in your institution?
Pardon? If animal are not available, IBC what? Can you go once again?
Sir, she's asking, is it necessary to make IBC if there are no animals available in your institution?
Yes, IBC is only for the chemical or the substance used for the, even as in hospital, most of the hospital IBC is there because so many chemicals are there who are hazards in nature. So they have to take permission from them. Even some in vitro studies, even some microbiological studies, some histological study, they need some this approval from IBC. So IBC is necessary. Yeah. But yes, if you want to use animals, so ultimately you have to take approval from an issued animal ethics committee. If it is lower, rodent in nature, but if it is big, you have to route it through IAC to the CPCs.
Any further queries? So thank you, sir, for elaborating on our responsibilities in providing specifications that will enhance animal well-being, which again pave way to advancement of biological knowledge relevant to humans and animals.
We now have a young medical graduate with entrepreneurial links in medication education, Dr. Mansi Bavkar, to talk about the use of computer-assisted learning in animal experimentation. Over to you, ma'am.
Hello, everybody. Good afternoon. My name is Dr. Mansi Bavkar, and I work in the field of medical animation. So we use technology to make medical education more engaging, more innovative. Majority of my work is in the field of animal experimentation, and I'll be talking about the same today. As the speakers before me have very thoughtfully articulated, animal experimentation is a complex issue, which needs to be dealt with a lot of nuance. On one hand, we have the lives of millions of innocent, feeling and thinking beings who deserve to be treated with compassion. On the other hand, we have medical research and innovation, which has saved billions of lives and continues to do so. Could the solution lie in adopting newer technology? According to my experience, there is one area of animal experimentation where technology can provide a genuinely viable alternative. That is animal experimentation for student education or for teaching. Let's see how. To start with, let's see what are the goals of animal experimentation in teaching. The first is to allow the students the opportunity to observe the actions of drugs on different organs and systems, as well as the interaction of drugs with each other. Secondly, would be to understand experimental procedures, the steps, do's and don'ts. And lastly, learning about the experimental equipment that is used to perform these procedures. All these objectives can be achieved using virtual animal experiments. Let's see how. Let's start with a video that visualizes guinea pig ileum dissection and experimentation. We can see the instruments used and their individual parts. We can see the dissection being performed. This is a small video clip to give you an idea. The actual video has a description, a voiceover, and a detailed explanation of the entire procedure of this experiment. We also show the graph and the mathematical calculations that help you arrive at the conclusion. The video ends with a short explanation of the relevant clinical theoretical concepts like the histamine receptors and how they actually act. Now I'd like to show you quickly how we can have students actually perform experiments. We'll be using the rabbit eye experiment to see this. So using our software, the student selects an animal and starts preparing the animal. Is this visible? Can you see the application?
Yes, ma'am, it is visible.
Okay, great. So the student selects the rabbit. The first step would be to trim the eyelashes. So use a scissor, trim the eyelashes of the test eye and the control eye. Then we measure the pupil size. So here it looks to be about 0.7. And here as well, 0.7. Then we add the test drug in the test eye and normal saline in the control eye. We note the baseline pupil size in both the eyes, that's 0.7. And your animal is ready to perform the experiment. Now you take the same animal and perform the experiment. The student has to identify the action of the drug and the timing of the onset of action. So that's why the student can perform the experiment at different times. In the interest of time now, we'll directly jump to 20 minutes. So here you can see that in the control eye, the pupil size has remained the same, it's 0.7. But in the test eye, the pupil size has increased, it's almost 1 centimeter. Now we'll check for the corneal reflex. Corneal reflex is present in the test eye and in the control eye. Now the pupillary reflex. Let's see, it's present in the control eye. We can see the pupil constrict. Then we check for the test eye and we can see the pupillary reflex is missing. I'll show it to you again. Control eye. See the constriction of the pupil. In the test eye, no constriction of the pupil. The student notes down all these points. So the pupil size in the test eye was 1 centimeter. Control eye is 0.7. Light reflex was absent. It's present in the control eye. And corneal reflex was present in both. So the student can perform this experiment at different times to find out the onset of action of the drug, and then eventually view all the information at a glance. Using this information, the student has to come to the conclusion as to what is the action of this drug and identify the drug. So as you can see, we get a good picture of how the experiment is conducted using both the video and the interactive module. So there are some advantages to using a simulated experiment or computer assisted learning, CAL as it is called. The students get an enhanced understanding of experimental concepts. They can actually understand what the action of the drug is by observing its effect on the animal. They get to actually perform the experiments and learn through doing. They can learn about the equipment used. These softwares are also cost effective compared to maintaining an animal lab. Also, they can be repeated as many times as needed without any additional preparations or cost. It is my understanding that we need doctors who are not just capable, but also kind and empathetic. This is only possible if their colleges and their curriculum displays the same empathy. So this is my personal inspiration for the work that I'm doing. These are my two cats, Olive and Mushroom. It's very easy to justify or excuse the sacrifice of animals if you've never been around an animal. But once you interact with any animal for a significant period of time, you can see that they can think and feel just like us. So any effort to reduce the unnecessary suffering of animals, according to me, is a worthwhile endeavor. And that is what my work is all about. So with that, thank you for listening. And if you have any questions, I'm happy to answer them.
Any queries that anyone has?
Is there any queries? You can ask ma'am directly.
Aparna, I think, has raised her hand, I see.
Hello.
Yes, ma'am.
Hello, ma'am. Ma'am, you are from Medimation?
Yes, I'm from Medimation Education.
Yes, ma'am. Okay, we have taken one CL software, but it is not from Medimation and from other company. The problem with Medimation, I think, is that it is highly costly. And because of the cost only, our management has denied it, and we have taken another from another company.
Is it?
Yes.
Actually, we do have like a cost-effective module. If you want, after our presentation, you can get in touch with me. We can see what we can do.
Yes, ma'am. And it was very nice seeing the rabbit's eye experiment. Very, very informative.
Thank you.
And I hope that this module would have been in our institute, but unfortunately, our management has denied it. But still, if in future, if possible, I will try, and you can provide it to us.
Absolutely. We'll definitely work with you. Yeah, I'll be sharing the number and email address of the company so you guys can contact us. Just a second. Huh? So we can always work with, whatever financial parameters of the college and come to some resolution to provide a maximum students with our experiments.
Thank you, ma'am. Ma'am, there is one more query that can you show experiments on guinea pig ileum?
Yeah, I have the full experiment. I can show it, but time-wise, if that's okay.
Yeah. Okay, ma'am.
So should I go ahead?
Ma'am, there are some time constraints, ma'am.
Yeah, that's why I showed a concise version. If you share your number, I can send you a link or something.
Yeah, ma'am. Sure, ma'am.
Thank you.
Thank you, ma'am, for introducing us to this truly innovative and interactive platform for medical education.
Thank you.
Thank you. We request Dean of Pharmacology at Amrita Institute of Medical Sciences, Dr. Pg Nair, to give her personal remarks on the issue of animal experimentation in medicine. Over to you, ma'am.
Hello, everyone. I am faculty alumni pharmacology from Amrita Institute of Medical Sciences, Kochi. I'm sure you all are very, aware today about animal experimentations in pharmacology. after Dr. Vikash Medhi and Dr. Dikshit, Ajay Prakash sir, and Dr. Manasi's excellent presentations. Very good. A take-home message from this, we understand, it is that animal experimentations are unavoidable in drug development. You cannot do otherwise but animals. And COVID-19 has taught us a lesson that we do require animals. Old timers like me, at least, we realize that the present generation of students do not know to handle animals. We should overcome that. And I am sure with Dr. Manasi's experiments, we will also be able to learn handling. I mean, the PG students of today will be able to learn handling of animals. And I'm sure these talks have given them a lot of knowledge. And I was very happy. Personally, I was very happy to learn a lot of things from the developments that has taken place. So many new ideas I have learned, and wish you all the best for all the students. And I'm sure that these topics will be very useful for your academic career. Thank you so much.
Now, I would like to request Dr. Princey Louis Palati to present us with the concluding remarks.
So friends, I'm sure this 2 hours has been very, very lucrative. And if you are a resident going in for your MD exams, you have 10-mark answers there in each of those 20-minute talks that have come through. And that was one of the main reasons that we have had this kind of a session today and brought out the relevance of animal experimentation. Imagine a forest. Imagine a forest. What do you think when you just imagine, close your eyes and just think of a forest? What is the animal that comes to your mind? Monkeys jumping across the vine like a Tarzan, right? Jumping across. Those animal, that monkey is caught and put in a little cage in the biomedical primate centers in UK and elsewhere, and many other places such as those, where they are kept in steel cages. And if they have been injected with, say, dengue or some other COVID virus or any other such, they will be kept in a double boxed chamber, and they will be experimented on for yours and my sake. And that is why we have such a long life expectancy today. Medical research, development of drugs has, gone on the backs of the animals for us to bring. But as we rightly know, Mahatma Gandhi has said this: the greatness of a nation and its moral progress is understood only by how that nation treats its animals. And we in India, we treat our animals well, and we know our culture brings it into us, and that is why we have this soft spot, and that is why we have a strong CPCA and a PETA. But friends, I'd like to draw a point that many of this generation doctors, if you want, our MD specialists who are already in, many of them have never touched an animal. I think I am 55, and my genre of people and beyond, earlier to me, we are the last few, should I say, extinct species, okay, who have done animal experimentation. And doing that, it's really bad, because your whole MD exam, forget what you are, but if my cat died, I am dead. Okay, so that was the state of affairs, and that's not to be, but we can learn and do many more from that. And the present system is good, and we have animal handling techniques, but we should, as a medical MD pharmacologist, you should know animal experimentation, because you may be called on to help in the drug discovery and in research using animals. And if you're not touched an animal, what will you do? How can you come out? Okay, that's my only one point on this side. Yeah, I'm a devil's advocate. I have to put both the points forward. I know animal rights activists may be not liking what I say, but it is for them and their lives too. Each animal rights activist, you're a human, you're alive and well because of the medications that we have. But of course, and with yours and the same feeling that we also espouse, we should see to the animal rights too. We are with PETA, we are also the people for ethical treatment of animals. You're not against. Okay, so blending this two, I think I should conclude. We have had a wonderful section right from Dr. Vikash Medhi taking us through what is the relevance and how it is. And we had alternatives taken by Dr. R.K. Dixit from KGMU Lucknow, and that was a wonderful session too. And we had Ajay Prakash to top it all with the type of ethics that we should know. In fact, friends, if you're doing animal ethics study or it's your thesis topic, or you want to do a research, the first block hindrance is the IEC is a hindrance, but AEC is another bigger hindrance, is what we think. And I thought, okay, I'll do something on Madaka fish. There also, the question comes in. We use 50 embryos for seeing embryo toxicity. They didn't allow me to do on something that was being repeated. So there it is. So we have good animal ethics committees, and we have to bring out good research. So friends, I hope it has been an interesting and good experience for us all to have. And last but not the least, we had this young medico, and she has brought to us Medi Animation. And before that, we had Dr. M.A. Ravindran, wonderful person who brought out in India the ex-farmer with all the animal experimentation done so beautifully, and we are really grateful to the likes of such to bring out a alternative, and that is also being done so well. And not only animal, we have simulation, and we can have also written out clinical scenarios where you can have like this kind in computer based simulation for also human kind of illnesses. So friends, thank you all, and I hope it was a pleasant day. Thank you.
Now we have Dr. Sanita Kuriachan, Associate Professor in the Department of Pharmacology, Amrita Institute of Medical Sciences, an avid researcher dabbling in clinical pharmacokinetics, to propose the vote of thanks.
My humble pranams at the lotus feet of Amma. It gives me great pleasure to propose the vote of thanks on behalf of Department of Pharmacology, Amrita School of Medicine. Gratitude is a powerful catalyst for happiness that converts that we have into more than enough. Our gratitude is to be expressed to our charming and considerate principal, Dr. Colonel Vishal Marwaha, who has graced us with a smile for stimulating our venture into the CME conduction. I thank our renowned stalwarts of pharmacology, our speakers, Dr. Vikash Medhi, Dr. Ragesh Kumar Dishit, Dr. Ajay Prakash. I would also like to thank Dr. Mansi Bauka for exposing us to animal experiments without animals. I would also like to thank Mr. Nitin in helping us organizing this event, and Dr. Pg Nayak for gracing this occasion. I thank each and every one of you from the bottom of my heart for sharing your valuable time with us. The logistic help obtained from the IT and Medical Illustration Department was phenomenal, and I thank them for all the help. Thank you all once again.
All of you have joined today will receive a certificate on your registered email ID. So kindly fill the Google form which is given on the chat box. I request all you to specify your name in the form so as you wish it would be on the certificate. So we are grateful for this opportunity to be your host for the day. We shall now conclude with the national anthem. Thank you. Thank you all. So please fill the Google form which are provided in the chat box so that you all can have the certificates. Thank you.
Thank you so much.
Thank you so
We'll
much.
conclude the session now. Thank you so much.